everybody. We're going to get started shortly. We had a little bit of close scheduling for our speaker. But this allows me to give you some information on how and why you should join the DNA sequencing research group. One, we're the best research group. Don't tell anybody else. Two, if you are here, that means you probably like genomics. So just to give you an idea, the last couple of studies we did, let's see, this year's, is a On-Bead Library Prep. Last year we did 2 years ago we did a methylation study. We've done a single cell study. So we've been working on a lot of studies in the last couple of years. We're working on putting all the publishing out, hopefully this year. Yeah, and so we're always looking for new members, because we're all really great people. And raise your hand if you're currently a member of the DSRG. Raise your hand if you were also a past member of the DSRG. Okay, all right. So we're a good group of people. I'm going to try to stretch this out because, like I said, there was some close scheduling, and our speaker will be right back. I don't need to share with you what he's doing though. So. Is anybody have any questions for me about the DSRG? Please? Yes. Oh, how to join. Yeah, so you can email me, you can flag me down, you can, we don't really have an email address, or you could message on the community. You can find us on the community. You can email Sarah or Mike, raise your hands if you know them. Or Ken, or just email Ken. Ken knows all of us. Ken will definitely get you there. So, welcome back, Sean. Thanks, Jess. We meet every other Wednesday at 2 p.m. Central. So, yeah. What, Sarah? Oh, 3 Eastern, yeah. Well, I don't, yeah, okay. Well, anyway, so we're gonna start with our talk. It's To Bead or Not To Bead, On-Bead Library Prep Miniaturization. It's being presented by Sean, who is currently at the University of Texas in Austin. And yeah, he's gonna give us your talk today. I'll let you explain the talk. So.
Good afternoon. Don't worry, I'm just as sleepy as everybody else from a food coma. But I'll venture to try to be as engaging and delightful as I can. Thank you guys for coming out. Thank you, Molly, for the introduction. Our DSRG's been really busy. We've been doing a lot of big work, a lot of big projects with methylation and our single cell benchmarking study for fixed workflows. So the past couple years have been pretty simple, and this one is pretty straightforward, so we'll dive in. But first I want to give a big shout out to all of our DSRG members. Molly's already kind of pointed out our new upcoming co-chairs, both Mike and Sarah. So if you guys are interested in joining the DSRG, we'd love to have you on board. But a big round of applause for Molly and for Fred, who couldn't make it this year, who've been carrying the DSRG for the past couple years and are ready to hand it off. So library prep miniaturization. Just out of curiosity, who's doing library prep miniaturization in their course? Anybody? All right, pretty popular topic. So we're all pretty familiar, and for those of you who aren't, the challenges around library prep miniaturization. Because we're miniaturizing in some cases 1 to 110, 1 to 120. I heard today that somebody's doing 1 to 150th. There are a lot of challenges around input concentrations. You still need to put the same amount of DNA in, so you just have to make it a lot more concentrated. You're handling extremely low volumes, sub-microliter volumes on the order of 10 to hundreds of nanoliters. And it often usually requires specialized liquid handlers to get into that range. But the benefits are huge, right? You're going to save a lot. What you're saving on reagents, you could be making in labor staff, fees, and all that. So we were kind of trying to brainstorm this year of how can we address some of these challenges around library prep miniaturization using the tools we have. And we thought maybe spry beads can come save the day. Maybe they can be our hero for this. Credit to Copilot for the beautiful little comic of our spry bead hero. Something we're all very familiar with. Everybody who's used NGS nowadays are using spry beads and ampere beads to their cleanups. And while this might seem interesting and novel, we're not the first ones to consider this idea. Back in 2011, the Broad set up a scalable, fully automated on-bead prep for doing and sorting whole exome workflows. And they had some really compelling and interesting results. They were inputting about 3 micrograms of DNA, sonicating, and then loading on to ampere beads, doing a cleanup with ampere beads, and then leaving the beads in solution during their on-bead, their end repair, A-tailing, ligation, and PCR steps. And they had found they got better yields from those workflows with the bead intact, and they didn't see any major changes. They even tested low inputs down to 100 nanograms and still didn't see any major shifts in mapping or calling rates for their exomes. So we said, well, we piggyback on that idea and take it maybe a step further. Maybe we can use spry beads to concentrate our samples, and instead of thinking of loading input in terms of volume, we're loading the physical beads, the input into the prep. Along with that, by keeping the beads in there, we're able to rebind the beads rebind the DNA onto the beads in solution. And so instead of adding spry beads every time you do a cleanup, we're just adding a PEG sodium chloride solution. And then on top of that, this also helps us to address the issue of low volume handling, because we're not having to elute off beads and transfer with a miniaturization workflow. We can just keep adding and adding and adding, and then just taking off the larger amount of the full master mix, and then just add the next master mix directly on top. So we came up with this idea, and to kind of test the feasibility of the concept, we took E. coli genomic DNA. We wanted to go a little lower than what the Broad did, so we went down to 20 nanogram inputs for this. And we split it between 2 kits. One, the Watchmaker Genomics DNA prep with fragmentation, which is an enzymatic fragmentation prep. So not doing sonication like the Broad did, we're adding in enzymatic fragmentation into the workflow. And we also wanted to test Ultra Express. The reason for the Ultra Express and the Watchmaker and not maybe say the Ultra 2 is we wanted to look at the kind of canonical 2-step post-ligation cleanup approach for DNA preps and a more highly optimized, highly streamlined commercial kit such as the Ultra Express, which really doesn't have any bead cleanup until the very end. The beads are just sitting around throughout the workflow. We sent out samples and kits to 3 different sites for each technology, and each technology was going to do 3 essential experimental groups. One was going to be our control, which we're going to follow the manufacturer's guidelines and protocol. The next was going to be an on-bead prep, where we're keeping beads in the reaction from start till finish, but not changing any volumes yet. And then the last was going to be a 1/10 miniaturization. And I say miniaturization, a lot of us who've ever done it, for those of you who raised your hands, a lot of optimization goes into it. The data I'm going to present today is no optimization. It is a straight 1/10 dilution across the board with no optimization on the adapters, indices, or any of the workflow. We just really wanted to see at that bare bones, what does it look like, and does it look like something that we can form into a more robust, repeatable prep? Once all these libraries were done, we sequenced them on the Illumina MySeq i100 to get back and aim for about 30x coverage for each sample. So I'll go over the protocol a little bit. And our control workflow for the Watchmaker, we're going to start off by grabbing the genomic DNA with a 1.8x cleanup on our ampere beads. This is allowing us to get rid of the volume there and just put the beads directly into the workflow. It's kind of moot because we still will resuspend in the recommended volume that Watchmaker has for the kit, but it's essentially going to be part of our control as we leave the beads on there for the on-bead workflow. But for the control, we do the 1.8x cleanup. We'll resuspend, and because it's the control, we're not leaving the beads in. We'll pull that off and transfer into a new plate or tube where we'll perform the enzymatic steps and follow the protocol as set. Watchmaker has 2 ampere bead cleanups, one post-ligation and then one post-PCR. For the on-bead workflows, both the on-bead standard and on-bead miniaturization workflow, we still do that 1.8x cleanup, but unlike the control, instead of eluting off into a volume and transferring the volume, we're just going to add the entire master mix, whatever water or TE Watchmaker recommends, directly to those beads and just resend the beads in our fragmentation master mix and start from there. And the beads stay in with that master mix throughout ligation. When we get to that post-ligation cleanup step, instead of adding more beads in, we're throwing in PEG with sodium chloride to bind the DNA back onto the beads at the same ratio, same volume as you would in the kit. Go through PCR amplification, we'll do our cleanups, and then again, at that elution step, instead of eluting and transferring, we just throw the PCR master mix with the properly diluted amount of water the kit says, or TE the kit says, directly on top of the beads, mix the beads really well, and move into PCR amplification. And same thing for that final PCR, post-PCR cleanup, we were rebinding to those same beads with PEG and sodium chloride, and then we're eluting in the standard volume. Same thing goes for the mini. The only thing that we didn't do a 110 on is the final elution. That gets eluted off in the same volume as the rest of the protocols. For the Ultra Express, because it's so highly optimized, and those of you who've used it know how crazy optimized it is, the only time we got a chance to add the beads was with that 1.8x cleanup, but we'll do the same as the Watchmaker. The control will get that 1.8x cleanup, and then we'll elute out and transfer it into a new tube, and then continue with the rest of the prep. On the on-bead workflow, we'll do that 1.8x cleanup, and instead of eluting and transferring, we add the master mix directly to the beads and leave them in through the fragmentation and repair, A-tailing, and after ligation, and PCR amplification. And at the very end, where we would do the cleanup and the control, for our on-bead workflows, we're just adding in the appropriate amount of PEG and sodium chloride to rebind. It's important to also note that they have their own bead reconstitution buffer, which will bind back onto the beads in the standard protocol, and we kept that part the same. We didn't change much of that, keeping to this high kind of no optimization run. So we did out through all the sites. We sent all back the data, and we started looking at initially what do the libraries look like? Does anything tell us in that sense? So here we're looking at Watchmaker. We looked at total yield for the entire library in terms of nanogram mass amounts. And here you can see on the graph to your left, we have the 3 different sites along with our total average of all sites for our control bead and mini. And while we see variations between sites, if you look at each site as a whole, we see that they tend to be pretty consistent. What we're looking at, if you were to kind of aggregate all of our sites together and average them out, is that in terms of total yield on the Watchmaker, we are seeing on-bead, same volumes as the standard workflow, have slightly higher yields for the libraries, and our miniaturization, which is being eluted in the same volume as the regular protocol, we're getting slightly lower than the control, but still more than enough to sequence forward, and everything is in nanogram amounts. For the NDB Ultra Express, we see actually a little bit of a different story with the aggregated total yields from all sites. The control seems to outperform in yields compared to the bead and the on-prep. And again, I'll restate this multiple times in the presentation. It's without optimization, but we're still getting plenty of yield from our on-bead and on-bead mini workflows to proceed on with sequencing and QC. But we're so far okay. We're getting library, we're getting yield. What does it look like? What does the average insert size look like with library shapes? So for the Watchmaker, here we're looking at average library size based on bioanalyzer traces. And we are seeing mostly pretty consistent sizes if you look to the graph on the right of the average of all sites. There is a good amount of variation, which is not unexpected, and more variation in the mini than the mini and the on-bead than the control. But we are getting pretty much around the same insert size, give or take about 50 to 100 bases. So we're still getting good full-sized whole genome libraries from these. The NHB Ultra Express, again, was a kind of an interesting different story, whereas if you look at the control and the bead workflows, we have similar sizing ranges, but for the mini, we have a much larger average size. This was an interesting concern from the NEB team. They thought that doing this mini bead approach may influence the fragmentation, and that could be a area for optimization. But like I said, for this study, we're just kind of doing the first glance optimization. So we're getting good libraries, we're getting good insert sizes, more than enough usually for whole genome sequencing, especially for E. coli. So next is, let's do sequencing. We ran this in the MySeq I100. We used the 25 million read flow cell, aiming for hopefully 27 million. We got something like, I think 28, so we just barely got it. And we were doing this on a paired N150 cycling conditions for the whole genome library. After which we analyzed it. For today's meeting, we did our kind of initial analysis using the Dragon small WGS app directly on the MySeq. So what'd that look like? So what we saw for the control bead and mini in terms of reads confidently mapped to the genome, we saw a pretty tight concordance both between sites and overall of well over 95% gene mapping, which is great. Blow you away, but at least we're not seeing any changes from adding the beads or adding beads and doing miniaturization to achieve this. So then we wanted to ask, okay, what about unique fragments? Maybe we're jackpotting something by adding beads. Maybe that can be influencing it, or with the miniaturization. So we looked at the percent unique reads for each one with the same analysis. And again, we're seeing similar concordance of at least between the on-bead and the control, the percent unique reads are pretty close. We'll see a lot more variation in the miniaturization protocol for these, but again, pretty close. I wanted to put a disclaimer out there. This data is only reflecting the watchmaker workflow. The NAB workflow, we had an issue with sequencing, and we're still kind of figuring out what happened and how we're gonna fix that. And NAB's been great on getting it up and running. So we'll have an update with this data with the NAB side of it too. But for the most part, from what we can see and what we are seeing on the flow cell, the story remains pretty close, where even on the Ultra Express on a miniaturized workflow, we're still seeing around this 85-ish%, 86% of unique reads and over 90% confidently mapped. That's the majority of the data that we have right now. We're hoping to continue doing analysis on this to kind of normalize mapped coverage across all the samples, and then maybe start looking at number of unique reads. When we start looking at SNPs and indels within the mapping rates, we want to take a look at the genome landscape of these samples to see if we see any dropout based on GC content throughout the genome, to see if there's any region there. But so far, it looks like this protocol at just doing nothing but swapping reagents out is holding pretty consistent to what the standard workflow would look like. We're working still with NEB to get those libraries analyzed and get that data added to this data set. But the implications of this is really exciting. This prep miniaturization, this 110, was done entirely on the bench by hand. No fancy liquid no specialized liquid handlers to handle the volumes here. And like I said before, we're not making adjustments to concentrations to pipette. We're just adding and going with the workflow. But we're excited for that. We're excited that this could be a method of using spire beads to increase concentration, allowing us to keep input the same across miniaturized workflows. We are seeing similar data that the Broad saw, that we might be getting higher recovery when beads are eluted directly in master mix rather than eluted and then transferred. And this lends itself to possibly being an automation-friendly avenue to miniaturization. By not having to have beads in a reservoir, mixing beads and transferring beads, you're just adding peg and mixing the beads directly in the plate. So making these a little bit more streamlined. The other cool thing is, whereas with the watchmaker, you had to swap plates after the post-ligation, you didn't have to do that with the on-bead workflow. Everything was done in the same tube through and through, without having to optimize any further than that. And so that's a really cool idea of being able to do more with less deck space. Along with kind of this concept of automation streamlining, fewer bead dilutions, fewer needs for plates, hopefully fewer consumables, and no need for those on-bead reservoirs and mixing them and making sure that they're not settling and ruining everything because it didn't get mixed well. Big thank you to all the sites that participated: University of Florida, Van Andel Institute, Boston University, Duke, University of Albany, Texas A&M, and the University of Texas at Austin. The people there are fantastic. This project was a slow crawl from when we kind of started, and as we started to learn more, there's a lot of optimizations that were done small scale. And so these people are amazing because data, the libraries and samples were sent out at the beginning of March, and we got data back last week. So it's been a real quick turnaround on getting this stuff done. So it's really exciting to see, and a big thank you to any of you that are here. I'm so sorry, but thank you so very much. And a big, big shout out to our sponsors: Watchmaker, NEB, IDT, Illumina, and of course ABRF, without whom we wouldn't be able to do this kind of studies, do any of our kind of studies, and bring you guys this kind of data. With that, I'm gonna leave it. If anyone has any questions, feel free to ask. If no one has a question for me, I have a question for the audience. Has anybody done on-bead preps in their lab or doing them currently now in their lab? Just out of curiosity. Yes? What application? Okay. Do you do it through the whole workflow of ChipSeek for the Excel, formerly Excel flow? Okay. Not as yet. Yeah, it's just one boom, boom. Yeah. I think we're also interested in testing out different bead types because everyone uses different beads. We did Ampere XP because that's what we've seen in the literature, and I think there's some interest in maybe trying different bead manufacturers. Did you use Ampere XP? Okay. All right. Well, thank you guys very much. I really appreciate it. And again, big shout out to DSRG for getting this study done.
Okay, all right. So our next part of our session is going to be an update on our 2024 or 2025, sorry, study, which is we kind of did a survey of core facilities, not just in genomics. We did a whole bunch of core facilities, and Sridar is here to present that. Thanks, Sridar.
So this was a long study. If you remember, if you did the survey, you'll definitely remember it. It had like 75 questions or so. But trust me, we're not going through 75 questions. It's been consolidated. It was some of the findings were neat. So we had about 41 respondents, complete respondents that we could find. Most of them, when we looked at the survey, they were either lab directors or core facility managers, and the rest of them were the lab technicians. Again, two-thirds, 70% or so were the genomic scores. Again, ABRF has been genomics heavy, DSRG, DNA sequencing, so it's not surprising that we were, but we did have responses from other microscopy and mass spectrometry cores, just not enough of them. One of the questions was, whether your core is service-based or whether it's just a walk-up core that people come in and you just utilize the instrument. Almost 90% were service providers to their institutions. When you look at the staff, most of the respondents were about average-sized labs. There were about 5 to 6 people in a lab as the median size. Though there were some that were larger, more than 15, and I think I can identify who those people might be just knowing the people at ABRF. When you look at the average sample volumes that were, or the samples that were processed, roughly about 10,000 samples or so was the average, though there were some of those large cores that were doing like over 100,000 samples a year. But on an average, regardless of the size of the lab, most of the labs were supporting about 100 PIs in their institutions or neighboring institutions. So that was good. The impact of ABRF and the people who work in the core facilities, even though it doesn't get recognized, is large. Then we started asking questions about how do you QC your RNA, how do you QC your DNA. So this is all consolidated. The fact that people are QCing is a good thing. And most of them tend to be the qubit. Some people are using the fragment analysis on the bioanalyzer of the tape station. And some people are doing qPCR. My lab does all three. So I was surprised to say that some people don't use the qPCR method to look at their libraries or their, their lab. So the qubit by far is the most commonly used QC metric, and that pretty much everybody seems to be using qubit, so that's good. Okay, then space becomes a challenge. Freezers, expense on the freezers and monitoring and maintaining them. So one of the questions we had was, okay, your project is done. Do you hold on to the libraries? Do you hold on to the starting sample if you have any left? And the survey says that pretty much core facilities hold on to both samples and the finished libraries anywhere from 6 months to a year, mostly 6 months. So do we need to hold on to them? That is something that we still have to consider because, I mean, once you give them the raw data, do you want to, are they going to come back to you in a year? Or if they come back, do you want to save your freezer space, which is as things are getting more and more expensive? Just give them back their sample. We ask them if they are going to do something in the immediate future. If not, then we actually give them back their sample. So, or unless we're doing a follow-up qPCR or some other assay with those samples, we hand it back to them here. Thank you. Here's your data. Here are your samples. So it's less inventory management for us. Then we looked at how these different size labs are if these different size labs are using LEM systems or some sort of billing methods or electronic lab management software. And by far, it's still being done by Excel. So Excel and notes and doing the manual way is still more popular in the 41 respondents that we had than others. And then I can see that depending on what their funding situation is, cost is, whether the data integration works well with their particular institution's financial management software, this might be the easier path of least resistance, even though it's more work. But then iLab is the second other the choice. People who were using LMS systems for managing their projects, where most of them were iLab users, and then there were some other commercial LMS that people were used. Cores rarely adopt any one particular system because they're constantly evolving, and So these things, they might be using them now, and then some other technology comes in which is not fit in. So this is something that's an ongoing requirement of, your limbs is not your buy once and you're done for whatever you want to use it for. These slides would be shared, so if, don't worry about the pictures. In terms of equipment management strategy, yeah, you QC the samples. We buy these fancy instruments, and then we have to maintain the instruments too, right? And on an average, instrument service contracts range anywhere from 10 to 20% of your capex expenditure, depending on the technology and how frequently it can break down, whether you can fix it yourself, whether it's your nano drop versus your sequencer, right? What are you more comfortable willing to fix yourself or hang around and wait for the service engineer to come, or the cost that is associated with it? So these are considerations that was targeted by the questions that were asked. And when you look at the most important thing that I was shocked at is the fact that there were some labs that reported that they had no strategy for maintenance. That is worrisome, right? Whether you have a service contract, whether you have an annual PM model, whether you have, okay, it breaks down and we will call in the service engineer, or we have somebody who can fix it in-house. All of those are fine, but the fact that some labs don't have any strategy, that's kind of worrisome to me. Because if you don't trust your instrument, if you don't validate and calibrate your instruments and make sure they're on a working basis, all that expensive data that you're generating, in this day and age, everything is going onto the open science clouds, and the rigor and reproducibility is getting more important. If your instruments that can be trusted, it's scary. But for the major instrumentation, it looks like most of the people have service contracts or have annual PMs at least. Some of them, about a third of 39% or so, we're doing service as needed. Costs. It's getting harder and harder to do core science in academia. Funding is getting tight, so I can see how these responses are valid. Some of them, they're just doing critical because it's expensive, but it's not as it's expensive to have the person come in and do it when your instrument breaks down, rather than have a full service contract all through the year, or you learn some of the things and just have them come in and do the other parts, like your mass specs and stuff. There's some things that can be user done if you know how to do it. So in our university, actually, the guy who runs the mass spec, he's a genius. He can take it apart, put it back together. Not everybody has that talent, but even he has challenges. And then, so there are some things that are covered, the critical services that are covered by service contracts for his instruments. The minor instruments, nobody has service contracts, but they know how to, they do calibrate and they do fix it in-house, so that's good. Cost consciousness and resource management. So how do we optimize our core facility budgets, resources? That's again not surprising. There's a strong emphasis on cost efficiency without compromising quality controls across facilities. The majority safely stretch budgets by utilizing unexpired leftover reagents. Right? So if you buy a 24 pack and you only run a 20 sample reaction and you got these 4 reactions left, people are instead of just throwing it away, are trying to scrounge up and use those things, as long as the reagents have been validated and we know that they work. So no one's compromising on quality. And miniaturization, as we've heard in many talks, including Sean's presentation just a few minutes ago, in terms of trying to reduce the volume of your reactions and maximize what you get out of that one kit. Even this goes back even to the Sanger sequencing, right? A lot of us do 1/8 reactions than what is recommended, and we still get enough beautiful data. So, and when it comes to NGS and stuff, the reagents are expensive. So if you can get more out of it, Stuart Levine from MIT had presented many years ago, pre-COVID, where he used the mosquito to do 1/8 reactions of his library preps What that allowed you to do is not just, it's not just about saving the money, but now that you save the money, you could do more biological replicates, right? So you can do science properly with the same money that you were going to spend, and then, okay, I don't have the money, so I'm just going to do duplicates. So that's good that the cores are trying to do those, miniaturize the protocols to save on the reagents and maximize on the science end of things. Using expired reagents selectively, well, we all know that what's on the label is not necessarily, it's not like, okay, March 31st, 2026, if that's the expiry date, on March midnight, the reagents just flat out die. That doesn't quite happen, right? Even with your drugs. But the industry has tested its stability for a certain period of time, and that's what they're allowed to put on the label. So maybe a month, maybe a month and a half. Sometimes flow cells and arrays, microarrays, I mean, if they're stored properly, you can take them 2 years later and they're just fine. Nothing's happened to them. So instead of chucking it, people are trying to reuse them and try to, or not reuse, I mean, use them and see if they work. But there's still quality control involved to make sure the data that you're getting is still, appropriate. And there's about 20% that only use new kits per project. Right, so that's the choice to make. If you're using an expired reagent, do you charge the PI the same rate, or do you tell them up front and cut them some slack with that? So that's a choice to be made. Troubleshooting, when shit happens all the time in cores, and so how do you deal with it? Communication, trying to make sure that you are upfront about it. Right? It's not about defense, right? This is a partnership. So when things go wrong, you troubleshoot internally, figure out, which step went wrong. Was it at your side? Was it something to do with the sample? Was it something to do with the process? So identifying that, 63% of labs said that they do that. The quality issue, they asked the user to repurify or reprep the samples to meet the quality standards. So if they were checking and initially things worked out or things didn't look good, you send it back, the student back and say, can you get? But there are cases where I'm dealing with a biopsy sample or I'm dealing with an experiment that some poor graduate student has been working on for 8 months and now here's my sample, right? They're not going to be able to go back and derive new sample. What can you do with that? So trying to work with it and communicating with the PI and the grad student that expectations, what are the expectations you should have of the sample? It's not that it can't be used, but not everything, you're not going to see everything. So you still try to make lemonade from lemons. And then the quantity issue, if you run out, what do? Well, most of us don't have a problem reaching out to the PI and saying, do you have any more? Sometimes they do, sometimes they don't. But don't hold back. Keep that communication line open, right? It's okay if mistakes happen. Mistakes can happen from any end. What happens if there's a new protocol, right? Do you have a budget to adopt the new protocol? Do you wait for somebody else to do the new protocol before you adopt it in your lab? Do you validate those protocols in-house before you bring it in, or you just trust the vendor's specs that are presented? So about half of the labs said they validate first and before offering it to their PIs. Adopting new protocols as a service, again, about half of them test it out with the free kits and stuff, and they adopt the new service as a new protocols into their service. And knowing all the people in the research groups, we are not risk averse, right? But it's not like we jump in and take protocols randomly because there's that validation time that we all require. We want to make sure it works in our hands. And if we don't, that's okay, because we know enough people in the community who are doing these things, or who have validated, or who have the budgets or the staff to be able to do these new protocols. And we refer the projects to our friends, right? And there are about 10% or so of people who, 5 out of 41, so it's not a good statistically significant sample, but they just simply say, no, we can't do it. You're on your own. That's okay too. I don't like to say that. I would like to say, no, we can't do it, but you can go to Molly's lab or Sean's lab or Scott's lab or whoever it is who's offering that service. Help each other out. And many thanks to the vendors who send us these free kits to allow us to do, or give us heavily discounted kits, to be able to test out these protocols and give us the time of the day with their technical people when we run into roadblocks. Data storage and retention policies. Okay, so there were a bunch of questions that talked about how long do you keep the data? What kind of data do you keep? Do you keep the raw data? Do you keep the analyzed data? Do you keep the intermediate data? How do you store the stuff? Do you keep it on hard drives? Do you keep it on institutional servers? Do you put it up on the cloud? And there are multiple models that are used by different people. About 40% or so of the facilities keep the archived raw data for more than 2 years before proceeding with deletion. At my institution, I say 6 months, but I've never quite implemented it. They don't know that it's there, but they assume that it's gone in 6 months. And so we offer it to them, we get an email back from them, they have the data, and then we are actually, technically, we are free to delete the data if we wanted to, unless they wanted to have an arrangement to do something else. But it's not uncommon to have somebody come back 5 years later and say, hey, you know that project I did, can you please get me this data? And so, and I know some people have deep storage that they charge for, going and bring it out of the archive too. Data charging policies, about 8 to 10% charge extra to store the data longer or to retrieve the data. Some don't. Okay, so key takeaways and operational best practices that we found from the survey: standardize the QC. Most of them do a qubit first, followed by a bioanalyzer or tape station and qPCR. In terms of retention, we need to formalize the retention policies. Are we going to keep your sample, keep your libraries, keep your data? How long are they going to be stored? Have that agreement in your protocols and policies of sample submission, so it's all clear up front. Your project management software, LIMS systems, even though a lot of people are starting to adopt it, because it comes with a cost unless you're building your own in-house systems. And Excel is still working. And now with AI and automate tools and everything that is there in Microsoft, it'll be interesting to see, from the talks that went on yesterday, how people could streamline some of those with the free tools. And academics, we are cheap, right? So we see what comes out of ABRF members who solve that problem. In terms of the instrument maintenance, please do have some form of coverage for your instruments. Please do calibrate your instruments, whether it's your pipettes or your nanodrop. It doesn't cost much to do that, but these are your tools. So that 27% that said they have none, goes out to them. But most of the other people have their instrumentation covered. In terms of the protocols, again, at least half of the people are willing to try out the new protocols and adopt them into their service portfolios. In terms of the data policy clarity, 6 months seems to be 6 months to 2 years is where data seems to be archived. Anything more than that, they're either not doing it or charging extra to do it. So these were the findings that we had. But if you have other questions that we should be asking that we didn't, let us know. We'll send out another survey. And I think it's important for us to have this communication even within our own communities and see what the pain points are and what the solutions that are out there that we could use. And at least try to document it somewhere, maybe even in JBT or something. So a new person coming into the field, starting a core, at least has some guidelines to follow, something to fall back on. Questions? Yep. All right. Thanks, everybody.
so we finished early. Yay, we can all squeeze in a nap before the keynote. Is that next? I don't know. Not gonna lie, I haven't looked at my schedule yet today. So we finished early. Enjoy the extra 20 minutes we gave you. Thank you for coming.